Okay.
You can touch on your title. The philosophers of science sometimes talk about the spirit of doing science. Really have that gift giving. So this is not a particular gift of philosophy.
I wouldn't say about my poor efforts, but
Yeah. I'm going to have a seat.
all right.
Say it again? Oh, good, thank you.
Okay, thank you.
Good morning, everybody. Welcome to our Department of Psychiatry Grand Rounds. It's a great turnout on a cold, rainy, not rainy anymore day, but preacher likes it cold, knows they're going to stay. We're really excited about this morning's Grand Rounds. I'm going to hand the microphone over to Sam Barnes in just a minute to do the formal evaluation, the formal introduction. Please fill out your evaluations for the CMEs. That was the other thought there. After the Grand Rounds today, there'll be a question and answer session in here, and then at 9:30 for trainees across the hall in the conference room. And so I'd like to hand over the microphone to Sam Barnes to do our introductions.
Good morning. It's my great pleasure to be able to introduce Dr. Karl Deisseroth as our Grand Round speaker this morning. Dr. Deisseroth received his undergraduate degree from Harvard University and his PhD and MD both from Stanford University, where he is currently the D.H. Chen Professor of Bioengineering and of Psychiatry and Behavioral Sciences, and he's also an investigator at the Howard Hughes Medical Institute. Dr. Deisseroth is best known for discovering and developing revolutionary techniques such as optogenetics and clarity, which his lab uses to better understand the neuromechanisms regulating adaptive and maladaptive behaviors in relation to psychiatric disorders. For these discoveries, Dr. Deisseroth has been the recipient of a number of awards, including the NIH Director's Pioneer Award, the Pearl Prize, the Breakthrough Prize, and the Mastery Prize, amongst many others. And today, Dr. Deisseroth will be discussing how the discovery of microbial proteins has impacted neuroscience and psychiatric research.
All right, well, thank you. Thanks for waking up early, coming to this talk. This is pretty early for neuroscientists for sure, even for psychiatrists. I still see patients, and it feels early to me. I focus on treatment-resistant depression. That's my clinical specialty. I see patients about a half day a week or less. But I also run a basic science lab, and so I'll be mostly talking about work from the lab today. But if any of you have questions about how this relates to the clinic, that's something I'd be happy to engage with in that question period. And I'll try to do a couple things in the talk. First of all, to give a really up to the moment, even some unpublished data, perspective on what's going on in the lab. And to do it at a few levels, try to make this point in a few different ways, how amazing it is that single-cell algae like this cute little being here, Chlamydomonas reinhardtii, which is a algae that has flagella and swims around in response to light, that a protein from this organism has helped us understand the brain-wide function and dysfunction of the brain in a really surprising way. So I hope to just capture some of that as well. Parts of it, I realize it's a broad audience. Parts of it will be, I think, technical. Parts of it might have some clinical perspective, but hopefully all of it will be at least a little bit interesting. Now, what we're trying to do is understand how the cells that make up nervous systems and their activity patterns, this constellation of activity, how this relates to brain function. It's not enough, though, to just see a flashing light that's a neuron. You'd like to know, you'd like to have that linked, if possible, to anatomical information. You'd like to know, oh, this particular cell here, well, what is its, what kind of cell is it? What's its neurotransmitter phenotype? What's its wiring pattern? So linking these two data streams is valuable. And then, of course, you don't want to just observe naturally occurring activity. You'd also like to play in activity as well and see if it's causally important in driving behavior. And so this is the technology that we call optogenetics, which is a way of actually providing patterns that were observed and testing their causal significance. And we've gotten to the point where we can even do that in the same cells and the same animals. Now, this particular strategy, one of the things that's amazing about it is it does depend on this microbial protein, which is in fact part of a broader family. Many single-celled organisms make these proteins. And there's 7 transmembrane proteins like many important G protein coupled receptors. They have embedded within them a light-absorbing molecule called retinal, all-trans retinal. It looks like vitamin A a little bit. And these were identified first in 1971. There's different kinds of them. Some pump a proton across the membrane in response to a photon. Some pump a chloride across the membrane in response to a photon. Some, like these channelrhodopsins from this algae, actually open a physical pore in the membrane. And they, of course, don't look quite like this. The pumps are a little more complicated. A lot of work's gone into understanding how the pumps work. The channels were somewhat of a mystery, though. This was the understanding in 2011. We wrote a review. There was some controversy, though, because there was not a clear precedent for how a pore, particularly a light-activated pore, could be formed within a 7-transmembrane protein. We thought that there was an internal pore. We knew that one of these helices had 5 glutamate residues, charged residues on it. But other people didn't think that. In fact, there was a prominent review that suggested that the pore must be formed by at the interface of a dimer or even a trimer of 2 or 3 of these monomers. But we suspected it was internal, and that did turn out to be right. Over the past 7 years, we've gotten a lot more information on the inner workings of channelrhodopsin. This has come from our crystal structure work, from our molecular modeling, and from a great deal of mutagenesis. And so, big picture is this is the internal pore that the blue arrows show, and it's absolutely lined with charged and polar, in fact, negatively charged residues. And this is probably why the naturally occurring channelrhodopsins at the time flux cation channels. There's a pathway that may exclude anions because of electrostatics and allow cations to go through. That makes it excitatory in neural systems, like many non-selective cation channels. This then would be stimulatory in a nervous system. And that turned out to be very useful. Here's the retinal in orange. It's embedded within this retinal binding pocket with all these little green residues around it. Knowing those turned out to be really important too. Why was all this important? Because we could start to play with the logic of this protein and fundamentally change it in new and interesting and valuable ways. So here's that excitation I showed you. We can flash blue light. We can put that gene into a neuron. We can flash blue light. And with one mutant that we made, this particular mutant here, we were able to make it a very fast channel. So we could even drive action potentials at 200 hertz or more because we were able to speed up this plant channel to that level. We also were able to make red light-driven tools. We found one that was slightly red-shifted from a multicellular green algae called Volvox carteri, and by 2011 we'd turned that into a red light spike driver, and that involved mutating this residue as well. Then we were able to create a bistable action. Instead of flashing one light and getting one spike, we were able to bias excitability, so we could flip cells into and out of excitable states in a stable way. So we didn't have to keep providing light, just make the cell more excitable to incoming input into that state with blue light, out with green, in and out, in and out. And then finally, we were able to completely flip the sign of the whole thing, make it an inhibitory channel that fluxed chloride ions, anions, instead of cations. We did that by remodeling the whole inner lining of that pore that we had found and changing those residues from those that would facilitate cation flux to those that would facilitate anion flux. And so we made an inhibitory channel. And then we were able to create bistable inhibition by porting this set of mutations on top of that inhibition. And now we can flip cells into a refractory, non-excitable state in a reversible fashion. And so this illustrates some of the value of getting to know at the atomic level this microbial protein. Gives you a lot of power for what you can do. And a lot of this work was done in collaboration with a really wonderful team of colleagues and collaborators. I'm going to zip through a few slides just kind of illustrating big picture what these changes were. The ones that zipped, that made the speed really zippy, these were the cheetah variants. The ones that addressed some of these problems that we had noticed of these plant channels not being fast enough. And here are the bistable ones, mutating a couple residues near the retinal. And normally, the channelrhodopsin turns off very quickly after you turn off the light in 10 to 12 milliseconds. But these modified ones stayed open. And so that gave us this bistable behavior. And these are called the step function opsins. To get the anion channel, to get the inhibitory version, we had to get the crystal structure, which we did in 2012. And it is indeed a dimer, actually, of two 7-transmembrane proteins. But the pore is not in the middle. Each monomer has its own pore next to the retinal. And we were able to look at that and see that see exactly where all those pore lining residues were. See that, as indicated in red here, that there was a negative electrostatic potential. And we were able to mutate that until it was largely positive, as shown in blue. There's a whole bunch of different mutations, modifications guided by our structure. And that let us create this inhibitory channel where we could block spiking with blue light. And then we were able to port the bistable mutations on top of that and create this bistable inhibitor, as I mentioned. There's a whole alphabet soup of different names for all these, which I won't dwell on. I like switcher. It's step waveform inhibitory channelrhodopsin. That's an example of the fun we have with names. All right, now the red light turned out to be really important. The red light driven version, but not for the reasons that we initially tried to go down that path. My initial motivation was to create two independent channels of control. I wanted to have to control with blue light and red light two separate populations of cells at the same time. And you can do that now. This is that multicellular green algae that we were able to do that in 2008. Feng Zhang, who was a grad student in my lab now, much more famous for other things, but back then he was an incredibly brilliant chemistry grad student in my lab. He found this red-shifted opsin from Volvox carteri, and you know it looked. So here was the first blue one that we knew about, and then in gray was the naturally occurring Volvox one, and we were able to shift it a little further with some mutations. And so there was this whole band in amber that had not been previously accessible. In theory, if you knew enough about the retinal binding pocket, you could, by yourself, try to shift the spectrum. And we can, but only a little bit. We're not smart enough yet to really powerfully shift the spectra as much as nature can. So we can shift it 10 to 15 nanometers by affecting the retinal binding pocket. But this big shift still requires to build off naturally occurring opsins. So we don't understand that as well as we'd like, but we understand it well enough to make red light driven spiking. And this first version, it was called C1V1. This was good for a number of reasons. It allowed single cell control, the first optical single cell control in vivo. So here is an interesting experiment. This is a living mouse. This was done in 2012 with Rohit Prakash and Ofer Yizar. We bring in a patch pipette and we record from a cell in cortex, neocortex of this living mouse. And we use 2-photon illumination to scan a plane of light just above this single cell, and we don't get a spike. And then if we go down into the cell, we do get a spike. And just below it, we don't get a spike anymore. So, and that's in the Z, similar thing, even better in X and Y. And so we can get single cell control in vivo, which was a big deal. And now you can do all optical experiments where we flip, and this is why the red light turned out to be even more important than we had thought. Some of the best readouts of activity are blue light excited. So that's the real value. You can now use the red light opsins to play in control and the blue light calcium indicators, like the genetic encoded calcium indicators, to collect your readout. So you do all optical play in and readout. That's what's really taken off. Together with David Tank, we were able to show in 2014 that you could do this, and it's actually a lot of fun. You can co-express both, C1V1 for red light control and GCAMs for blue light readout. And you can just hop from cell to cell with spots of light. We and others have developed various ways to deliver single or multiple spots of light across your population. You can target individual cells. You can ping them with light. You can see their responses, see how you're doing, see local circuit responses as well. This is just individual cells indexed on the y-axis and time on the x-axis, and intensity of the calcium elevation readout of activity in pseudocolor. And so you can pick cells by virtue of what they're naturally doing during behavior. And so here's a case in visual cortex where I wonder if we can dim the lights in the front a little bit. But here are, whenever this red asterisk is flashing, these two neurons in the red boxes are responding. And here we're guiding light with a spatial light modulator to just those two neurons that we picked because of their orientation selectivity. So now you can guide cells by virtue of their naturally occurring patterns. You can do that at the single cell level. You can also do that at the population level. Here's an experiment. Tina Kim in my lab developed a fiber photometry method where you can have a single fiber optic like the ones that we normally use to deliver optical excitation, and you can do that but also collect the readout light through that very same fiber in that same animal. And this is very valuable. Here she's co-expressed a Volvox-derived red light-driven opsin and a blue light calcium indicator, and is targeting the dopamine neurons, the ventral tegmental area dopamine neurons, very involved in many roles in behavior, including in reward and motivation. And what she was able to do was first, give this mouse, this living mouse, a reward, a sugar water reward, and in light blue, she could collect that naturally occurring signal in response to reward from the BTA dopamine neurons. And then she could, in that same animal, same positioning, she could try to provide that pattern optically. And so here's her first attempt, and she can see she's coming on too strong there, but she can back off in light intensity until she exactly matches in that animal the naturally occurring pattern in those same cells. And that may seem simple, but I think you appreciate there's a lot of value in that to be able to match or modify, as you will, the naturally occurring patterns. And so this is what we can now do with modern optogenetics. With the various mutants, we can be acute or chronic any time scale we want. We can excite or inhibit. We can have multiple fiber optics across the brain, target cells or cell types or projection types. And we can match or modify the timing or amplitude of naturally occurring patterns as we like in the animals. So that's a very quick overview of where things stand with the technology. Now I'm going to take one of the several forays into experiments, applications that relate to things I think most of us in the room care about, which relates to clinical conditions, but also the natural adaptive correlates of those conditions. Now, many of you will know that Helen Mayberg and her colleagues, as well as others, with observational work in patients with depression, have found patches of elevated activity in frontal regions of the brain, including, in this case, subgenual cingulate gyrus. In patients with depression, elevated activity, often correlating with symptoms of anhedonia, reduced experience or motivation for reward. And this observational work is complemented by other interesting findings. For example, artificially elevating activity in prefrontal cortex of rats can also lead to reduced seeking of reward. And this is shown in various ways. Anto Banchi and his colleagues in 2013 elevated medial prefrontal cortex activity in cocaine-addicted rats and found that that alone could powerfully suppress cocaine-seeking behavior in these addicted rats, which was quite a striking result. So much so that they followed that up with a clinical study in human beings, in cocaine-addicted people, using TMS to elevate activity in the prefrontal cortex, and they reported in the first paper, and Ento says now it's holding in larger studies, not yet published. There was, in those cocaine-addicted people, a profound suppression of cocaine use. And so that is interesting. Of course, it needs to be replicated and extended. But big picture here, all of these indicating elevated activity, in some cases, in some contexts, in prefrontal cortex, can suppress a reward experience of seeking. And I had a student in my lab, Emily Ferenzi, who was quite interested in this and wanted to see if she could build a clinical bridge based on this concept and try to understand at the brain-wide circuit level what's going on. And to do this, to make this bridge, to form this bridge, she wanted to do fMRI and to do it in rats. And so she worked very hard on habituating rats. They would lie still in the scanner without habituation. Even if their head fixed, there's a lot of wiggling, but she was able to really solve that with a very painstaking process of habituation. And she could get beautiful, for example, visual fMRI bold signals. And now the exciting thing is bringing some of these tools I told you about earlier, in particular these two, the fast red excitation tool and the bistable excitation, and help those form this bridge to the clinic. And so here's what she did. So first, she wanted to use this fast red one to ping the dopamine system. She wanted to have a consistent, causally evoked indication of what happens when you ping the dopamine neurons, what happens in the brain-wide circuit dynamical sense, what is the circuitry that's recruited by this stimulus, which we showed was indeed rewarding, appetitive, and highly motivating, in fact, for the animals. And so this is what she did. She was able to stimulate the ventral tegmental area dopamine neurons. If you just express yellow fluorescent protein in those neurons, you don't see a response. If you express channelrhodopsin variants in the dopamine neurons in these rats, you do get interesting brain-wide responses that were particularly prominent in the ventral and dorsal striatum. We knew this was rewarding for the animals because they would lever press many hundreds of times per day for that light to the ventral tegmental area dopamine neurons. And the more an animal had bold contrast in the ventral striatum, the more lever presses it would exhibit. Then that was with one opsin. That was with a fast red opsin, just to ping the reward system and do something that we knew was rewarding in a highly precise way and see the recruitment of reward circuitry and quantify it. But then she also wanted to provide this clinical-like intervention, this elevation of this stable elevation of activity in medial prefrontal cortex. And so she picked the bistable step function opsin, blue light driven, so compatible with the separate red light experiment in the same animals. Expressed that in the medial prefrontal cortex of rats, and again, with yellow fluorescent protein, there was no response to light, but she could see this patch of elevated bold activity in the step function opsin expressing rats. And she did some interesting experiments. Not only did this reveal itself to promote an anhedonic set of behaviors, there was reduced preference for sugar water compared to regular water, there was reduced social interaction and other interesting findings. But then she could now bring these two opsins together in the same animals and do brain-wide imaging and ask how is there an alteration in the dopamine neuron recruitment of reward circuitry in response to prefrontal cortex modulation. And so this was the experiment, two fiber optics in the same rat, one to medial prefrontal cortex, one to target the dopamine neurons. And we know how these two opsins work at a detailed level. And it was pretty striking. Instead of the, when she turned on the medial prefrontal cortex opsin, the recruited signal went from this to this. And so there's a powerful suppression. And this is interesting. It's a bit of a meta intervention. A change in the excitability of one part of the brain is affecting a conversation going on between two other parts of the brain, the VTA and its dopamine neurons and the downstream reward circuitry. And this showed us, and this, by the way, also proved to be an anhedonic type intervention. And so this gave us an example of how elevated prefrontal cortex activity could lead to a clinically significant finding like anhedonia in principle. And I think it's interesting, just to reflect on how, of course, we're just at the early stages of trying to understand this sort of thing, but this insight really comes from botany and from algae. And to help make this point, which I think is an interesting one, I want to show this picture. This is Andrei Fominzin. He was the botanist who first found the plant behavioral responses, the phototactic responses of Chlamydomonas. And he did a simple experiment. He had a dish of algae that he collected from the Neva River and shown mild intensities of light and saw that the algae would collect on one side of the dish. If he used brighter light, they would find an optimum. And he published this. He was Russian in St. Petersburg, but he wrote in German, and this was submitted to a journal with a French name, so very multicultural. So that I think it's an interesting story. And now I want to go even farther in thinking about adaptive and maladaptive behaviors and get into some unpublished work and a whole new branch of work in my lab that's using zebrafish and mice together. So depression, as we all know, it's not just anhedonia. There's a complex collection of symptoms. Other things people may think about when they think about depression, they may, or that they may experience. There may be hopelessness, profound passivity, and this is another thing that we can, like anhedonic-like responses, that we can find behaviors in animals that are suggestive and evocative of this sort of response. You all will be familiar with the forced swim test, which is can be properly described. Not only, of course, it's interesting, people use it in the context of identifying drugs and thinking about depression, but of course, you get interesting responses in wild-type animals, and you see a transition from active coping to the stressor to passive coping, which can be an adaptive response. It can be a healthy and reasonable thing to do. If you put a rodent into water, it will swim, try to escape for a while, but over a couple minutes, it'll transition to a more passive state where it's simply floating in the water. And what we chose to study was that transition. The transition from active coping to passive coping, to challenge, in wild-type animals, basically animals that have not stressed in any particular way. And to build on that, though, and then to start to add stressors on top of that. Now, Melissa Warden in my lab had done some interesting experiments with this preparation. She did a fiber optic intervention in rats. She was recording from prefrontal cortex at the same time, and she found a lot of interesting things. For example, she found, oh, by the way, here you can actually see that transition from active coping to passive coping. This is the animals kicking over time. You can see there are these bouts that an animal transitions to a passive state. Yeah, please. And she found, for example, there was a projection from prefrontal cortex to the dorsal raphe that favored the active coping state. She found a different projection to the lateral habenula that favored the passive state. So a lot of interesting experiments in these rats. But a lot of this was, you can go and test anything you want, but you have to, the way you do these optogenetics experiments is you inject a virus carrying, for example, a channelrhodopsin into one part of the brain, and then you deliver your fiber optic to another part of the brain to deliver light. And because the axons that go from here to here over a few weeks fill with the channelrhodopsins, you can recruit cells defined by having this connectivity with a virus injection here and a fiber optic there. And you can go and test individual hypotheses you may have. But one thing that was dissatisfying to me about this, not that I didn't like it, great work, Melissa's a great scientist, she's now at Cornell doing great work, but you're missing a brain-wide picture. So you're intervening in one spot, but you're not collecting brain-wide information on dynamics. And so certainly there's some level of insight, although you're collecting behavior, there's some missing information, I would say, to put it mildly, on what's actually happening in the brain. So this is why we have a branch of our work now that's in zebrafish, and this gets to the unpublished work I want to tell you about. So these are small vertebrates, and we do a variety of different things with them. One of them is this preparation. It's head fixed, tail free, so embedded in agarose. They are healthy and happy. They can stay in this preparation for many hours. I don't really know if they're happy. But they definitely flick their tails, and so we can see at least whether they want to flick their tails, I guess, which is something. And we can collect a huge amount of information. So we worked very hard on different strategies for collecting cellular resolution, brain-wide information. And we worked on methods I won't get into, light field microscopy, light sheet microscopy, but for true single neuron resolution, we stick with 2-photon imaging, and we have piezos that allow us to scan our plane of illumination rapidly through the volume, and we can end up collecting many tens of thousands of neurons, effectively the entire brain of this organism during various interesting behavioral exposures. And this is the sort of data we get. What we have is one of these blue light excited calcium indicators that are expressed in all the neurons. And then, you know, as I mentioned early on, we want to not just see the activity of a neuron, but we want to then go and find out more about that neuron if it's doing something interesting. And it turned out to be not trivial to do that. So Matt Lovett-Barron in my lab figured out how to make this alignment of data streams work. If you just look at your live genetically encoded calcium indicator as shown here, and then you take the fish and fix it and stain it, and you look at the fixed indicator, same fish, same indicator, red and green, they don't align very well. Yellow indicating where there's overlap, and that's just because, you know, with fixation, there's a lot of shape changes that happen. But what Matt figured out was basically a computational pipeline to align those two data sets at cellular resolution across the brain, so you can indeed come and find the exact cell or cells that were interesting to you during life, find them after life, and then do rich molecular phenotyping of those same exact cells. So a very interesting opportunity. We published this just a couple months ago. We call it multi-map, a multiplexed alignment of molecular and activity phenotypes. And we did a variety of validations of it. For example, here, you know, labeling some spinal projection neurons, fixed versus live, blue versus red, not very good overlap. That's before registration. Here's after registration. Sub-micron level precision after the alignment. So we know those same cells, and then we can start to do some very interesting brain-wide, global, unbiased experiments. We can ask very simple questions initially, and then get more complicated. For example, what Matt did in these experiments is he asked a very simple question: what are all the neurons across the entire brain of this vertebrate that encode or represent the state of alertness or vigilance? And how are you going to do that? Well, in the head-fixed, tail-free preparation, you can present visual stimuli in this looming dot paradigm. Looks like something, you know, coming down on the fish. It's not a good thing for the fish if that is happening. And so there's an escape response, which we can see by its tail. But what's very interesting, and you see this in mammals as well, there's a distribution of latencies to the escape response. Sometimes the tail flick comes really quickly. Sometimes it comes late. And you can ask, and this varies trial to trial. Some trials fast, some trials slow. And you can look at all the neural activity across the brain, you know, high activity in red, low activity in blue, and, you know, ask, are there neurons that predict whether there was a fast or a slow escape latency? And there are, and it was pretty interesting. So we were able to molecular phenotype dozens of neuron types, and there were some expected observations, which was good in a way, helped us validate the system. So the noradrenergic locus ceruleus cells, there was a reduction in escape latency when those cells were active, suggestive of a heightened arousal state. But other things that were completely unexpected, this very interesting peptidergic population in the hypothalamus, the CART cells, cocaine and amphetamine-related transcript, had an even more powerful representation of the vigilant state. Other interesting things, neuropeptide Y also. Here's one going the other way. Somatostatin neurons in the hypothalamus predicted a sluggish state, a long latency escape response. Okay, so a lot of information, a lot not previously appreciated, but global unbiased collection of information. And then we can go back to the mouse and we can ask, all right, these things that we didn't expect that we found, are these conserved? Do they hold in mammals as well? And we can go back and do that with the fiber optic-based methods I mentioned. This is our fiber photometry method that we described in 2014 and that Tina Kim updated and improved in 2016. And you can have animals, you know, in brightly lit rooms while you're collecting and providing activity to targeted cell populations. And so we had again a reaction time task, a Q, which is a tone, and a lick for water reward. And again, there's this distribution of latencies to respond. And if you look, and we used various Cree driver lines and so on, including you know, a CART, iris-2 Cree line, for example, to target those populations that were found to be interesting in the fish. And we saw a very striking correspondence between what we had seen in the larval zebrafish and what was in the mouse. For example, that somatostatinergic population, hypothalamus, showed this long latency response. The CART cells showed this a faster response representation of that state. Now, all this is correlation. How much is causal? We also came in then and did optogenetics through the same fiber to control these cells and asked if they actually modulate the alertness state. And the cholinergic and adrenergic cell populations that we've identified actually were causal in the state. The hypothalamic populations were not causal. Driving those or inhibiting those did not have a powerful effect on the actual reaction time. So we think the hypothalamic cells are, rather than causing the state, are using that state information for some other computation or reason that we don't yet know. But that illustrates now how you can go from fish to mouse in this interesting way. Now, that's published. I want to tell you about something unpublished now that is going from this very simple reaction time task to the active to passive coping state transition, still in fish. And so what Aaron Andelman and Vanessa Burns and Matt Lovett-Barron did was, this took many years. You know, you've got to, if you wanted to study this in fish, you can't use a forced swim test. That was the first barrier we had to overcome. So we had to create a situation where the animals were going through an active to passive coping state transition, and this has to occur at an age of the animal where where it's still small enough and clear enough that we can see all the neurons in the brain. And that was hard and took many years, but they finally got there. And this is what we do. We deliver 1 hertz, very mild but inescapable shocks across the tank to the animal. There's an initial active coping phase where the animal swims more, and then that transitions to a passive coping state where it swims less. A great many controls establishing the nature of the state. For example, it's not that they're just dead as one control. They fully recover, but over some tens of minutes. And it's a context-dependent recovery. That's only if they're in a new tank with new water. If you leave them in the same tank with the same water, they do not recover their former swim speed. A number of other interesting experiments, including this one, ketamine, the putative fast-acting antidepressant, slowed and reduced the magnitude of the passive state transition. Now, for the sake of time, I won't go too much into the task itself, but I'll go right to the findings. And so this is then the kind of exciting thing you can see. You can see all the neurons across the brain during a state transition like this, and you can ask in an unbiased way what's going on. And this is actually happening right before your eyes. This is a fish that's going through that transition. And this is a movie that's going to loop around and start from the beginning again in a moment. But you may want to pay attention to what's going on in these two lateral structures here. So here's the movie looping around, starting again. This is a fish that's receiving this mild electric shock stressor that it can't escape from. And what you're seeing is accumulation of activity in these two structures. It leaps right out at you. Was consistent. And what is that structure? Well, it's actually the fish homologue of the lateral habenula, which was pretty amazing. And here it is. So we've got, we segment many tens of thousands of neurons computationally. Each little white dot is one of those neurons, and we can see this steady accumulation of activity in the lateral habenula, much, much weaker in medial. What's interesting is in the raphe, and remember the opposite direction causality in the rat of habenula and raphe in the forced swim tests, contrast to this increase in habenula, we see a decrease in raphe during this state transition. Both of those effects blocked by ketamine, shown in magenta. Not much else happening across the brain, a little bit of reduction in dorsal thalamus. But then we can go and ask, that's an interesting correlation, is it causally important? And so for that, we can do some interesting experiments. So we can target Channelrhodopsin using Zebrafish Genetics to the lateral habenula structure. And we can optically drive it and optically record the brain-wide activity state, first of all, and then we can do behavior, which I'll show you later. So this is interesting. If you have a Channelrhodopsin negative fish, no Channelrhodopsin, and you shine light on one side or the other side of the brain, you don't see much of a response. If there is Channelrhodopsin in the habenula and you illuminate the habenula, you see some brain-wide responses, which are interesting, as well as the local response indicating that you've successfully stimulated the habenula. And so here's what we see. This is our first pass on the analysis. First couple seconds, if you stimulate the lateral habenula, you see an increase in activity in the lateral habenula, so that is good. Shows you're doing what you think you're doing. Not at the bulk level, not a lot changes across the brain. But 8 to 10 seconds later, we see a decrease in the raphe, which is very interesting. So we think that decrease in raphe could be causally downstream, because it certainly can be causally downstream of the lateral habenula activation. Now, those raphe neurons, what's going on? Here we can use a multi-map to get to know those neurons a little better. And we can go and stain, of course, the raphe, like all other nuclei, is complex, has many kinds of neurons in it, not just serotonergic. And we got a surprise here using multi-map. We found that it was the serotonergic cells that were immunopositive for 5-HT that by their activity more represented the early active coping phase of the response, and it was the non-serotonergic cells in the raphe, a little of that, but that had more of the reduction later. That's more associated with the passivity transition. So that was an interesting surprise. Even more interesting and surprising what was going on in the lateral habenula itself. And here's an example of that. There's a lot of dynamics going on here, but what you might notice, and what turned out to be the case, it's not as if all the habenula cells are ramping up in activity together at the same time. In fact, some don't even take up in activity until quite late in the stressor, and then they show this transition still remaining dynamic, but at a higher activity level. Others make the transition earlier. And so we were pretty interested in that. Is there a population representing a short duration stressor or a long duration stressor? And if you think about it, that's pretty important in deciding what to do. That statistic of the stressor is quite important in deciding whether you should have a state transition or not. But it wasn't quite like that. It wasn't that there was an early or late responder population. In fact, there was a completely smooth temporal tiling of habenula recruitment over the duration of the stressor. New cells ticking into this higher activity state over tens of minutes. That didn't happen in the raphe. With the raphe, there was some kind of threshold was crossed that we don't fully understand yet, and all those transitions happen roughly at the same time. But it's the habenula that seems to be representing this very important statistic of the challenge, and doing it by progressively recruiting individual neurons. And so a lot more interesting work to get into on that, which we're actively using multi-map now to get to understand those individual habenula neurons better. But you can also, causality is interesting from the point of view of the brain-wide dynamics, but also for behavior. What happens to behavior when you drive the habenula? And here, Zebrafish Genetics again, Channelrhodopsin in the lateral habenula structures. We did Talia Lerner in the lab did patch clamp recordings to verify that we were generating excitatory currents in these cells in response to light. And if you're a fish and you don't have Channelrhodopsin in your habenula, as shown in black, and you get some light, you zip around a little more, and that's reversible. This is speed, millimeters per second. But if you do have Channelrhodopsin in your lateral habenula and you get light, it's actually, you don't see that. In fact, there's a slight suppression. That's a small but significant effect. But those are naive fish where nothing particularly bad has happened to them that we know about. Here is animals that have been through this prior stressor, and there's a profound evoked passivity that's actually quite stable. That's exciting, providing activity, and we see the passivity. What about suppressing activity? Are you able to remove passivity? And here we can use a inhibitory opsin. And here's an animal receiving the inescapable stressor, active coping, transition to passive coping, and then yellow light, which drives this inhibitory opsin in the habenula, silencing the habenula. And the movie is pretty interesting. This, I'll play this movie. This is actually a fish representation here. It's right in this phase, barely moving at all. You'll see a couple little zips here and there, like right there, but generally not moving much. Then the yellow light comes on, and there it is, back in its active coping phase, seemingly instantaneously. And that's a reversible when you turn off the light effect. So this habenular activity appears to be necessary and sufficient for giving rise to the behavioral state transition. So this is the opportunity here to try to come to a deeper understanding of the causation of adaptive and maladaptive behaviors at the cellular level, and knowing a lot about the cells, not just where they are, but who they are. We're also working on making this sort of approach viable, although the ping-ponging back and forth from fish to mouse works well, but also we'd like to find in mouse those same neurons that we observed during life. And here, just the last few slides, one thing that's really helped is these hydrogel methods that really help us gain access to cells that we've observed during life. So hydrogel tissue chemistry, what is that? We described this in 2013 and 2014. Clarity is one version of this. Many other labs now have described different variants. The basic idea is you start with your cells within your tissue, and they have interesting molecules, proteins, and nucleic acids. And you build from within all the cells at once a scaffold. This is all done after life. You build in a new meshwork, a framework that you can then, through interface molecules, attach interesting biomolecules to, like nucleic acids or proteins. You can then do various manipulations on your tissue. You can remove lipids, for example. There are reversible size changes you can have happen. You can do ongoing chemistry. You can infuse in enzymes and do reverse transcription and so on. And basically you end up with the interesting biomolecules replotted onto a new coordinate system, effectively, that you can work with in interesting ways. So that's hydrogel tissue chemistry. This is what the initial polymer backbones we described were like. There's other variants that have been described now that are similar but have interesting variations on the theme. The chemistry that allows you to functionalize and then link either polypeptides or nucleic acids to the framework is pretty well now worked out. You can have different kinds of monomers that make your gel. These are the gel monomers that are in widest use, but you can have other variants as well. And those, through the interface or functionalized polypeptides and nucleic acids, allow you to link your proteins onto a gel that becomes something you can work with. Now, the unique features of this method that are particularly relevant for this exact question we're dealing with, cells, you observe their activity during life, what can we find out about them after life? Because it's an aqueous-based method, there are no organic solvents that are used. There's nothing that's going to bleach or quench the fluorophores. And so if you have a native fluorescent protein, like a GCAMP, genetic coded calcium indicator, that you've used to observe your activity, that fluorescence is preserved. And if you have multiple different GCAMPs, a red one, green one, blue one, those will be preserved as well, and that really helps in the registration process. You can do also many rounds of labeling because the new formulation is quite robust to staining and destaining and restaining and destaining. And you can look at proteins and nucleic acids. Here's an example of a mouse brain going through this process. So a lot of opportunity there, and we've in early work we've established nucleic acid-based methods. For example, you can look not just at translated proteins, but you can look at microRNAs. You can look at immediate early genes that will give you a trace of activity that had happened in those same cells. Here's ARC labeling, for example. And then in many cases, and again, this is very early, and not to take away from the value of the fish-mouse work, which I think is always going to be an interesting paradigm, but in many cases, we've now been able to show that you can go and find those same cells that are interesting. Here's a population of cells in prefrontal cortex, and we can find individual cells with known molecular phenotypes. Like this particular one is an NPAS4 cell in medial prefrontal cortex, and we know this particular cell type is recruited by appetitive cocaine stimuli in these mice, and we also can follow the wiring of that same cell across the brain and find that it progresses to the nucleus accumbens in mice. And this kind of thing works at the cohort scale, so we can find that PFC to nucleus accumbens cells are recruited by cocaine, less so by shock. The opposite is true of projections from prefrontal to lateral habenula, whereas the projections from PFC to VTA are roughly equally recruited by positive and negative valence stimuli. So this is sort of the what we're hoping to do, the path we're going down, and that's why we have fish in the lab now. It's a lot of fun. As you can see, everybody's very happy. And amazing group of students and postdocs who've worked with us, worked on this sort of thing with us over the years. Just to highlight Matt Lovett-Barron, who developed the multi-map approach, which is so central to where we hope to go in the future, and the other people I mentioned along the way. Emily Ferenzi is a neurology resident now at MGH, and all our collaborators and colleagues. So thanks very much.
Karl, thank you very much for that tour de force. I guess doesn't really actually do it justice. So the glimpse into the brave new world of the future of your work. I'd like to open the discussion now for questions from the audience.
Taking it to the clinic, would you care to speculate on the antidepressant effects of light treatment in the 450 nanometer range? It's primarily, we've thought, on melatonin, but there's new data that it may be similar to the photobiology in plants, replacing heme with chlorophyll and releasing nitric oxide. I'm just wondering.
Yeah, the question is, antidepressant effects of light in people. It's very interesting, very real effect. Obviously, you don't introduce opsins in, so what is the natural light receptor? What is the propagation of that signal? Where does it influence, depression-related circuitry? Very interesting question. So it would be nice to study that in animals to see maybe you could map out those relevant pathways. And I'm not an expert in that, unfortunately, so I don't really know what the status of that field is. But yeah, maybe it sounds like you know more than I do about it. I don't know where the pathways would enter the relevant circuitry. Yeah, presumably, there are pathways from the suprachiasmatic nucleus to through one or two synapses to monoaminergic nuclei, and that could be a plausible mechanism.
The other rapid-acting one, in addition to ketamine, is sleep deprivation, which some people think is dopaminergic.
Yeah, so yeah, the fast-acting effects, albeit transient, of sleep deprivation are extremely interesting, and those have replicated in animals, as you likely know. You can have after a night of sleep deprivation, actually both in rats and mice as well as in people. But also, I believe there's some early work in fish suggesting that's also true. So again, we might be able to find out the circuitry.
So in the BOLD study into the regulation of anhedonia by the medial prefrontal cortex, were you surprised to see the dorsal striatum rather than the ventral striatum light up as a site of modulation?
Yeah, so we saw both dorsal and ventral light up. Question is, were we surprised to see dorsal striatum show up as a site of that was recruited by ventral tegmental area dopamine neuron
And then in the data you showed where prefrontal cortex suppressed that, it was the dorsal striatum that seemed to be the site in the data that you put up.
Yeah. The dorsal, certainly that there was a powerful suppression of the recruitment of dorsal, as you say, but also a ventral, statistically overall, the experiments. So that's certainly true, and that was a bit of a surprise. There's less of a, one of the classical, features that define the prefrontal cortex has to do with its connection that may be more powerful to the ventral striatum than dorsal. And so we were surprised by that. We don't know the mechanism of that dorsal striatum interaction. We have followed up the ventral striatum work a little bit, and we identified a pathway, and we think we have a handle on how that works. Yeah, the dorsal, that's an interesting question. I don't know the anatomical substrate for that effect.
Great presentation. I really liked your finding about the activation of the lateral alveoli and how that changed activity in the dorsal raphe. And we found similar data in collaboration with Andrei Deravakian, how a stressor can selectively change the activity of serotonergic versus non-serotonergic neurons. And I was wondering in your experiment if you tried to inhibit the lateral alveoli and see whether that inhibition of non-serotonergic neurons in the raphe was actually changing or abolished.
Yeah, great question. I showed the effect of inhibiting the habenula on behavior. I didn't show anything relating to the serotonergic or non-serotonergic raphe cells. We haven't done that experiment yet, but we could do it, just haven't gotten to it yet. Yeah, that'll be a good thing to do.
Well, it's a crazy idea, but that's what you trade in with this amazing team that you have. With your hydrogel approach, you pretty much have stability of proteins, it sounds like, stability of almost everything. What would it take? What would you have to overcome to be able to allow action potentials to take place in that sort of a setting?
Oh yeah, I do get that question. It's not completely crazy because I've gotten that question a few times. Right now, it's really nothing that we do is compatible with ongoing survival of the cells in the hydrogel methods. But it's and a lot of what goes on. There's the proteins, nucleic acids are chemically modified. Lipids are disrupted. There's a lot that's problematic.
I just want to hear you say it's impossible.
It's impossible at the moment.
So as long as we're trading in crazy ideas, I'd like to hear your thoughts about the possibility of one day using gene therapy or using viral vectors to put opsins into human patients as a treatment for depression or other psychiatric disorders.
Yeah, good question. It's not something I'm working on in my lab, but people are thinking about it. There has been already a company in Texas has put Channelrhodopsin into human beings, into their retinas, as part of an attempt to treat a progressive form of blindness. And the idea is by conferring some light sensitivity to cells in the retina, you may be able to confer some sort of responsiveness to the environment. That, this is a registered clinical trial. I haven't heard that anything bad has happened to those first people yet, but I haven't heard any good outcomes either. So people are doing it, starting in accessible structures. I also think when we start talking about depression, we have many ideas for that kind of experiment from our work, but I think it's we really have to come to a much more deep understanding before we take that step for psychiatry. What's faster, I think, would be these optogenetics guided therapies, like the cocaine study I talked about earlier, where the causal knowledge of the suppression of cocaine consumption by an optogenetic precise intervention led to a precisely designed optogenetics guided clinical trial that seems to be working. That, I think, is the much broader set of possibilities, which is already working.
In your work about the differences in activity between serotonergic and non-serotonergic cells of the raphe in the active to passive stress coping as duration of stress increased. Is there any information about what the non-serotonergic cells are?
Yeah, good question. What are those non-serotonergic cells? We're phenotyping them now. We expect there will be some GABAergic and dopaminergic cells in that population. Personally, I'm very interested in the tyrosine hydroxylase positive population in the raphe, and I suspect some of them are in that group.
So in the vein of optogenetic guided therapies, the optogenetic guided therapies, like you said, overstimulating MPFC will produce anhedonic type behaviors, yet we use TMS to treat depression and potentially overcome anhedonia. How can you speculate? Are you overdoing it in terms of stimulation, in terms of the rat model, or is there a dose response or type of neurons that are being affected? Because it's inconsistent. You overstimulate and you produce anhedonia, and here we stimulate prefrontal cortex and we treat anhedonia.
Oh yeah, okay. So so general question, how it's of course not so simple that although it may have come across that way in the talk, it's not quite so simple that it's a general phenomenon that elevated activity in frontal parts of the brain will always give the same effect. I hope I didn't say that, although I might have implied it. I think there's a great complexity to exactly where. I mean, you consider all the real estate that's involved in the human frontal cortex. But even in the rodent, we know there are parts of medial prefrontal cortex that drive one kind of response and completely opposite response in an adjacent prefrontal cortex population. So I think there's a lot of spatial complexity and also the kind of stimulus that's involved is going to be important as well. This is part of the reason that it is kind of amazing that the cocaine study actually worked, because of how much more prefrontal cortex there is in human than the rat. I don't think most studies are going to work that well unless you've got a much, and this is why I was saying earlier, I think we need a much deeper understanding of circuitry before we really start to design human clinical trials, particularly those that would be interventional. I think we probably need, patient-guided, patient-specific therapies that are informed by that particular patient's symptoms, that particular patient's diffusion tractography, so we can see where the relevant tracks are, position our TMS coils, and in a very patient-specific way. I think that's going to be a big part of the future.
Question? Here you go.
I was interested in what you had to say about neurons that accumulate statistics of the environment. That's one way to put some of the things that I study if on a shorter time course. I was wondering, even on the time course that you're looking at, which is over many seconds, what sort of electrophysiological mechanisms your lab is currently thinking about? Do you think it's network level, just to put it broadly, like our favorite, EI balance and or do you think it's very specifically gated, like, right at the synapse, definitely kind of transmission?
Yeah, mechanism, network, and cellular mechanism of this, encoding of this state and the progressive recruitment of cells. Very interesting. And broad categories. Is it a network phenomenon, or is it a cellular phenomenon? Maybe there's a smooth, gradient of some potassium channel expression in all these cells. Same level of input, but different ones overcome that threshold with different ramping up of activity over time, depending on their own unique threshold. We don't know the answer to any of these things, but now we can do things like stain for channels, look for cell intrinsic properties that might suggest different excitability profiles and link those to those same cells' naturally occurring patterns. that's in the works. We're looking at channels. Then your network hypothesis, I think, is also reasonably likely to be interesting as well. And so we're trying to look at local wiring within the venula to see if there's different input connectivity to the different cells. Yeah, we're thinking and talking about this all the time, but don't know the answer yet.
So I'll ask one last question. So depression is an incredibly complex phenotype. And you've got that complexity in your physiology and anatomical work going all the way down from circuits all the way down to atoms. And yet the behavioral phenotype you're working on is essentially an escape response or a transition from active to passive motor activity. Where's the next level of the behavioral phenotype that you're going to want to pursue in these organisms?
Yeah, I mean, this is an open question. Is how much can fish do? We're and particularly in the head fixed, tail free preparation, you've got basically one kind of readout, which is a tail flick. How much can you really infer? How broadly applicable is this? Another problem with the fish is at this age, it's very easy to aversively condition them. They don't show a lot of appetitive conditioning, which is quite interesting. They still have a little yolk, for example, so they may not be as motivated by food rewards. They are free swimming. They will hunt prey and capture prey, little amoebas and paramecia and so on. And
do they show spatial preference or place preference within their swim environment?
We can do a place aversion, conditioned place aversion. We have not succeeded in getting conditioned place preference in fish at this age. So what we're working on though is we're trying to put together a panel of behaviors that we know that the fish can do, and for cases I completely would stipulate that the fish are not going to do everything we want them to do, and so that's why we're continuing to work hard on the mouse versions of this finding the same cells.
Any other questions? Well, Karl, thank you so much, and I know we're all very appreciative of the work you've presented to us and the information you brought to us. But the key moment is now going to be accomplished by our chair, Igor Grant. And
Karl, I'm first of all very sorry I wasn't here to welcome you. As chair of a department, you sometimes have to go to administrative meetings at 0700. So I very much apologize, but I did hear part of your talk, and obviously it's been a very lively discussion. Thank you so much for visiting with us. We do have a tradition, as Neil alluded to, and that is that we will welcome you as an honorary member at the UCSD Department of Psychiatry, and I know it's also sunny at Stanford, and so when you're out jogging or doing whatever outdoors, we hope you wear this with pride. I'm trying to open it up because you've got quite a bit of hair.
Well, it's actually just a big head. There's not much there.
There we go. See if it fits. Anyway, thank you so much.
Thank you.
minutes break, and then the trainees cross in the conference room.
And I'll say hi to you a little bit later.